I think, 12 presentations. So, okay, our first speaker is Logan Johnson, and he's coming from Iowa State University.
Cool, thank you. Hello. Oh, okay, sounds good. Hello, my name is Logan Johnson. I'm a PhD student at Iowa State. And as you've obviously probably seen by my title, I study meat science and muscle biology. So my talks may be a little bit different, coming at it more from an agriculture perspective and a food production perspective than maybe more a molecular and genetic perspective. But starting off just with a little background today, meat production is a large part of the Iowa State industry and also producing food for people such as a high-quality protein like meat is important, has a lot of nutrients and essential amino acids. But even today as consumers, we can go to the supermarket and find meat products that vary in their quality. And so what I mean by that is you could buy a pork chop that has a slightly darker color or a slightly lighter color, or it could be really tough or tender, juicy or not. And that variation is difficult to predict from a producer's side. And so one thing that we know from a lot of previous research looking at the structural component of the sarcomere, or the basic contractile unit of muscle, some of the degradation of those key proteins are really influential in depicting and influencing quality of that product. And so we have some soluble and insoluble proteins, with the soluble ones being in that sarcoplasmic fraction, and then the insoluble ones being in that thick and thin filament. And so degradation of those proteins we've linked to meat quality previously, but we're really interested in understanding specifically with this myofibrillar protein or that thick and thin filament fraction, some changes in relation to meat quality there. So to do this study, we collected some pork loins from a commercial pork plant. So these pork loins depict current genetics and nutrition and production practices. So we didn't really influence anything on that live side or the production side. So these represent a population of pork that you could buy in the commercial marketplace today. And we categorized them in evaluated quality, such as tenderness, juiciness, and some other factors I won't get into. But we had some that varied in their ability to retain moisture, or their ability to lose moisture. And I kind of depicted that here as well. So low being it lost a low amount of its weight to water or purge, of what you might see in a package sometimes, and a higher group that lost a greater proportion of its weight in water. And so we're really trying to understand what's different about the muscle tissue there between those samples. And to do that, we fractionated our proteins, and we really think this is advantageous to our analysis. So we can separate the soluble and insoluble proteins here, so that sarcoplasmic, or those soluble proteins, versus the insoluble pellet there. And so what we then did is we wanted to wash or remove any unbound or non-covalently bound proteins from that insoluble fraction. We then solubilized them in a urea buffer and then submitted them for TMT 10-plex analysis and quantification at our protein facility on a Q-Exactive mass spec. So from that analysis there, we identified about 428 proteins. And in this case, we're really trying to understand what's really different about them. So we're kind of in that discovery mode of really understanding future direction of research. So we were a little stringent in some of our cutoffs of determining differences. So we included only proteins that had at least 2 unique peptides or more. It had to be in at least half of our samples, or 38 of our samples. We then transformed and normalized that data and did a moderated t-test in R with the Lima package. And from that, we had about 20 proteins that were different. And so this table over here is proteins that were greater in the higher quality, that lost less of their weight in water. And so what we found is that a lot of these are really structural proteins or contractile proteins that make up that sarcomere, those thick and thin filament components of that. And then conversely, when we see differences between the high and low, so one that lost a greater proportion of its weight to water, we see a lot more metabolic enzymes there. And so that's really interesting to us, because those we wouldn't expect in that fraction. But that might indicate that they're degraded or oxidized, or some other modifications are occurring to them. And so ultimately, this work is really trying to understand, again, what's different about them. So hopefully this will lead to some new areas of discovery for us. We also have some analysis with that low ionic fraction, as well as some metabolomic data. So again, we're trying to work on building a greater understanding of some of that phenotype that's really impacting the quality of the product, so we can ultimately moderate our production practices and genetics to improve the quality of a product that people can consume. So thank you, and I'll take any questions.
Thanks a lot. Do we have questions? Yes.
Hi, thank you for the talk. When you filter from about 297 proteins to 133 proteins using the cutoff of if they're present in about 38 samples or so, or greater than 38 samples, does that mean that you lose the ability to determine if there's a peptide present or a protein present in specifically just a slice of your groups, your categorizations?
Yeah, that's probably one limitation to this. Again, we're trying to identify majorly abundant proteins, so we're not proteins that are more different. But that is something. Learning a little bit more about the imputation, I think, will help kind of maybe modify some of the analysis that we do here. But we're really trying to look broadly, and so really seeing proteins that are expressed more greatly or that are more abundant in these protein or in this fraction is what we're really looking for. And so we're less worried about maybe proteins that are less abundant because they might contribute significantly less to some of that ultimate phenotype that we're seeing.
Thank you.
We have time for one more question, if anyone has.
Nice talk. So from a sample preparation point of view, how are you actually characterizing water loss or retention? Like, how do you quantify that?
Yeah, that's a good question. So I didn't get into a lot of that, but what we do is we actually age these porcelains. So often you think of, you could hear dry or wet aging of samples like that. And so that allows some proteolysis to occur. But once that happens, then we would slice those porcelains into chops and then measure the amount of drip loss that would occur from that. And so that kind of simulates really the timeline of product flow from production to a grocery store. So that kind of is our simulation and measurement of water loss there, or weight purge loss, I guess.
Great. Thanks, Sultana. Let's welcome our next speaker, Casey Heaney. And she's coming from University of California, Santa Cruz.
Okay, so hi, my name is Casey Heaney. I'm from UC Santa Cruz, and I'm from the, let's see if this can, okay. Okay, so I'm from the Sanchez lab, and we primarily use imaging mass spectrometry to apply to a variety of applications. So you heard Allison's talk yesterday about squids. We also do ovarian cancer biomarkers, and I study cheese rinds. So why we think cheese rinds are interesting is because they're a really good model system for evaluating microbiomes. They have about 10 to 12 species, whereas other microbiomes can contain hundreds of species, so it's easy to study in the lab, and it's also very reproducible. So we're particularly interested in this penicillium species MB, and it contains a global genetic regulator called LAA. So global genetic regulator means it can control a variety of specialized metabolite biosynthetic gene clusters. So we want to see how that changes in the wild type versus the LAA knockout. So I can do that by doing fungal plug, like the Penicillium MB fungal plug on a Brevibacterium lawn. Brevibacterium is an actinomyces, also cheese-derived. And from there, do MALDI imaging mass spectrometry on our TIMS top flex. So to do that, I take a cut of the agar and put it onto a steel metal MALDI plate. And from there, apply matrix, and then put that into the instrument, which per each pixel, multiple spectra are generated and grouped into one. So I'm able to go through that spectra and see ion images, which reflect the relative intensity of the feature of interest. So I have here like actual ion images from so my experiment. So I picked feature 702, and you can see in the agar and Brevibacterium condition, it's not very present, but visible in Penicillium MB. downregulated in the delta Ly-A condition, and then also differential expression when co-cultured with Barbibacterium. From doing some data analysis in Cardinal, which is a really cool R package for doing IMS, I created some segmentation plots which show spatially distinct or statistically significant spatially distinct regions. And I can also look through those segments and see the corresponding spectra. So with even more data analysis in Cardinal, I can eventually find out some statistically significant features and hopefully answer the question, which is what specialized metabolites are under the global genetic regulator control of Ly-A, and what is the exact function of Ly-A. I'll take any questions. Thank you.
Great job. Thank you. Do we have questions? No? Okay.
Yeah, I'm curious, what's the nature of Ly-A? Is it like a transcription factor? Do you know what it's doing functionally? And are there maybe like lab strains of penicillin that you could use to molecularly look at this?
So you're saying, is Ly-A a transcription factor?
Yeah, like do you know is it a transcription factor? If not, how does it globally regulate these other factors?
Yeah, so that's what we don't know, and so that's what we hope to find out, and maybe through like figuring out what gene clusters it regulates.
Thanks.
Awesome. Let's thank Casey one more time. So our next speaker is Thiago Luiz Alves e Silva, and he's coming from NIH.
Hello, can you listen to me? Okay. Thank you for the opportunity to present my work. I'm studying in the interaction of the malaria parasite with mosquitoes. Malaria is the deadliest disease transmitted by mosquitoes, and every year it kills around 6,000 people around the world, most of them children in Africa. Before being transmitted, the parasite must invade the mosquito salivary gland. In the salivary gland, the parasite will stay like up to several weeks waiting to be transmitted to a new host. The parasite is transmitted when an infected mosquito bites susceptible rows, and the parasite is delivered in the saliva. So we can think in the saliva like a pharmacy, like CVS. It's because the saliva is full of proteins that interfere with hemostasis. So you have a lot of proteins that can inhibit inflammation, complement activation, and coagulation. Because of this, most of what's known about the saliva composition came from transcriptome studies. So essentially, in the transcriptome, you look for proteins that have a signal peptide and that are expressed exclusively in the salivary gland. And then you assume that these proteins will be secreted. So what I want to know is when you have an infection, how the parasite shapes the proteome of the salivary gland and the saliva. Okay, so this is essentially what we have done with our sample. So we in the MS2 we used data-dependent acquisition and CGI, and then we analyzed the samples using the MaxQuant and Perseus. So in the whole salivary gland, we saw a differential expression in several proteins across several protein families. One thing that was interesting is that the classical saliva proteins, from up to 60 proteins that you can find, only 4 were differentially expressed. But many other proteins that are secreted were differentially expressed. And overall, this proteome had a good correlation with other transcriptomes that were published, around 60%. So these results suggest that the parasite can impact the composition of the saliva. Okay, so what happens when you look to what happens, what's going on in the saliva. So when we looked in the saliva, we had a big surprise. We saw a very strong downregulation of proteins in the saliva. And in case anybody is wondering how I collect saliva, I take the mosquitoes and I put the mosquitoes to spit in a solution with a neurotransmitter. It stimulates the mosquitoes to salivate. Okay, so I collect saliva from 100 mosquitoes, and then I analyze these samples, okay? So which proteins are disappearing? When you look into the list of the proteins that are disappearing, it's very interesting. The proteins that are upregulated, they are essentially being depleted in the saliva. So then we asked why this was happening, and to answer this question, we performed IFAs. I made antibodies against some of these proteins. I'm giving here only one example. This protein here is the PPO6, is an immune protein. And you could see that in the infected salivary gland, here you have the parasite in green. These proteins accumulate on the parasite surface. So essentially, the parasite is trapping these proteins in its surface. So what we learned with this study is that.
So anyway, there's been a lot of methods that have been developed outside of the kits, but none of them have been really systematically compared to find a sort of best method. So for this experiment, we chose to work with the S trap specifically because it's kind of a commercialized, streamlined, fast protocol for protein isolation and digestion, and it's really easy to use. And we picked 6 methods to test and compare against each other. So I have this table right here. The first 2 are unpublished methods developed especially to go with the S trap. So we have bead beating and boil in an SDS buffer. And then these next 3 are all published. We have a 2-phase sodium hydroxide phenol extraction, a 2-phase SDS phenol extraction, and then an SDS boil with precipitation with TSA, or TCA. And then finally, a QIAGEN DNeasy kit. So the extraction methods were performed, and a BCA assay was done to determine the protein content. And at this point, the SDS precipitation and the QIAGEN DNeasy kits did not contain sufficient protein in order to move on to the S-trap kit. So they were axed from the study, and we continued on with our top 4 methods. Following S-trap digestion with trypsin, they were analyzed on an Orbitrap Eclipse with a single CV FAMES method with MS2. And then post-data processing, or data processing was performed with Proteome Discover, and then we also did some de novo sequencing using Peak software. So you'll notice that I have some values written. And so our Proteome Discover identifications, we had 205 total proteins identified. And all of these counts are very similar. There's some variation. We used 3 FASTA for each soil type. Our garden and sandy, sorry, I meant to cover that earlier. And then with our de novo peptide identifications, there's a bit more variation, and you'll notice that the SDS phenol has the highest counts out of the other methods, but counts aren't everything. It doesn't mean it's the best method. So we're gonna move on to these 2 figures. So the top figure is a PCA plot, and you'll notice these yellow dots that are kind of outliers to this main cluster, part of the SDS phenol group for the sandy type soil. And ideally with replicates, you want them all to be kind of clustered together. So that kind of acts as that method from the study. And then we can move on to the bar plot, and you'll see that there's a lot of variation in the sample abundances. And so the most consistent method for this was the bead beading method. So continuing on, our next steps that we've already performed, but we would be performing in a hypothetical scenario, is taking this best method and doing a robustness test with different sample volumes, and also trying to establish reproducibility. Yeah, that's it.
Thanks a lot. We have questions. I'll ask a question. So you mentioned that you've used a Ames device to do your analysis. Judging by your numbers, it doesn't seem like your mixture is very complex.
Yeah. So
Did you try doing the same analysis with no Ames and see how much Ames helped you?
no, but part of the issue is we only had the 3 FASTA files. So we had a very large FASTA file that we were using for the identifications, and it kept clogging up PD and it wouldn't run it. So we simplified down, we picked the top 3 FASTA files for each. So if we add more of a database for it to pull from, we would anticipate higher counts. And also we loaded only 25 micrograms of protein onto the S traps. So that could have been a factor as well.
Okay, makes sense. Thank you. So our next speaker is Jordan Berg from the University of Utah.
Okay. So I come from the Rudder Lab, where we get really excited about metabolic regulation and trying to kind of expand on our current knowledge of what the metabolic network is. And so we think a lot about the different levels of metabolic regulation that can exist, and all of the ohms can interact. But one, plane that we're really interested in is regulation between the proteome and the metabolome. And these form the basis of a lot of regulation at the enzymatic level. And you can think of like feedback regulation, where if you have too much of a metabolite, it might bind to an enzyme and shut it off for a bit so the cell can catch up. But identifying these interactions in a high throughput manner has been very difficult. And so something that's been developed in our lab is this Midas platform. Sorry, hopefully you can see what I'm pointing out with the laser, but this top right, where we have this plate where each well has a dialysis film. And so on one side, we start with isolated purified protein. The dialysis film the pores are small enough where the protein can't exchange through the chambers. But then on the other side, we have this metabolite pool with over 400 metabolites that are physiologically relevant. And we allow these 2 chambers to reach equilibrium. And so you can think if a metabolite is interacting with a given protein, it's going to be enriched in one chamber. And so after this, we crash out the protein and then feed these chambers through mass spec to measure what's the enrichment of each of these metabolites in the protein chamber. And so you can imagine that we're screening 400 metabolites versus I think we're at like 200 or 300 different proteins. And this is going to expand. I think you're in the process of building that metabolite library up to over 600 metabolites. And so the visualization challenge becomes an issue. And that's what I've really focused on with this project, is building a visualization and analysis platform to enable interpretation of this data set, or of these data sets. So this is called Electrum. And I think, so when we think about a metabolic pathway, a lot of the times we think of it very linearly, like substrate A is catalyzed by enzyme A and it turns into metabolite B. But we see that when we overlay the Midas data on the central carbon metabolism pathway, we see that it's not just this linear pathway. We see a lot of these interactions where, for example, here we're highlighting citrate synthase. And yes, it interacts with citrate, which we expect, but then it's also interacting with all these other steps in the pathway. And so we're working on following up on some of these hits and validating those. But then I think when we expand this, so here we're only looking at the metabolites that are annotated in central carbon metabolism. When we look at the entire Midas pool, we see that this metabolic regulation is extremely complex. And so some things that I'm doing computationally to follow up on these results. Okay, I'll wrap up. Sorry. So if we look at the enriched metabolites with a given protein, can we extract enriched metabolite substructures? And can we then use that in docking simulations to predict where the metabolite or how the metabolite is interacting with a protein? Does that indicate some function that that's happening or having? And then another aspect of this, to try to build on the cellular context of where these interactions matter, is scraping through PubMed using natural language processing to identify enrichments of, if we have a metabolite protein that's predicted by Midas, can we replicate that somehow in the literature, and is like a certain disease enriched when this metabolite and protein are co-mentioned in the literature? And so yeah, this is again called Electrum and Midas, and you can check out the preprint up here. I guess, yeah, the details are there. So, and I'll take any questions if we have time for that. Thanks.
Thanks. I see a question.
Hi, great talk. I guess one question I'm wondering, coming from pharmaceutical and small molecule, you have a mixture of metabolites, but how do you know then if a metabolite interacts with a protein, changes the structure that's representative of what's happening in the body?
Yeah.
Because you can imagine that it might not be representative because it's happening in a certain sequential fashion, but you can't control that sequential fashion in this.
Yeah.
So how do you overcome that and make it representative of a disease phenotype?
Right. Yeah. So that's very difficult. I mean, this is an in vitro model where it's not going to perfectly replicate that scenario. Some things that Kevin, who has spearheaded the Midas project, has done is, so we have to, because all these metabolites have different properties, we have to pool these metabolites in 4 different pools and run the experiment. And so what he's done is modulate the amounts of certain metabolites to see, do we still get the same, I guess, enrichment results from that?
I guess maybe what you could do is, if you had a control, you could take one pathway with no metabolites, see how that alters a protein, but then to see if that protein is altered in the same way if you include separate pathways for different disease phenotypes.
Yeah.
You know what I mean?
I think so, yeah.
Okay.
Maybe we can talk a little bit more about that.
No,
yeah.
I'm wondering about it.
Yeah. Yeah, I mean, there's, I guess, this is, in essence, a screening platform, too, where they've done a lot of, like, with these hits that are statistically enriched, done a lot of, like, thermal shift assays to show or demonstrate that it is actually interacting, as well as some other validation experiments.
Awesome. Thank you so much.
Thanks.
So our next speaker is Lauren Mazurkiewicz from University of Wisconsin-Madison.
Hello, my name is Lauren, and I'm a second-year PhD student in the Weeks Lab here at UW-Madison. And my project centers around the very unique post-translational modification dipthamid. And so dipthamid so far is only known to occur on one protein in the cell, elongation factor 2, a protein involved in translation. And it actually takes 7 different enzymes, DPH1 through 7, to synthesize this very chemically complex modification. And despite this modification being conserved amongst all eukaryotes, its precise function in the cell is not very well understood. It's been hypothesized that perhaps the modification plays a role in maintaining translational fidelity. However, there has been really no systematic cell-wide search for other dipthamid modification sites or for other functions of these DPH1 through 7 enzymes. And there has been more emerging evidence from recent genomic screens that some of these dipthamid biosynthetic enzymes might have other functions in the cell. And so the goal of my project is to identify functions of dipthamid and DPH 1 through 7 throughout the cell. And to do this, we thought that a great tool would be to use discovery-based proteomics. And so to start off, I have been doing TMT, quantitative proteomics, just to get a general idea of processes that DPH 1 through 7 might be involved in. So I labeled each of the DPH 1 through 7 knockout yeast strains with different tandem S tags. And I analyzed these samples using LCMS-MS and quantified the relative abundance of each protein in each knockout relative to the wild type. And from this, I was hopefully going to see upregulated or downregulated proteins that might indicate processes DPH 1 through 7 is involved in. And interestingly, I found that only DPH2 and DPH3 knockout cells had significantly upregulated or downregulated proteins. And when I looked more specifically at proteins that were significantly upregulated in DPH2 and DPH3 knockout yeast, I found that many of these were involved in processes related to ribosomal biogenesis, DNA damage repair, the cytoskeleton, and transcription and RNA processing. And so, based off of this, I hypothesized that maybe DPH2 and DPH3 have other functions in the cell unrelated to dipthamid biosynthesis. And in the future, I hope to use other mass spectrometry-based proteomic methods, such as maybe affinity purification MS or enzyme catalyzed proximity biotinylation to look at the protein-protein interactions of DPH2 and DPH3 and to better pinpoint how exactly DPH2 and DPH3 play a role in these broad biological processes that I've identified so far. And so thank you for your attention, and I'll take any questions.
Thank you. Do we have questions?
Yeah, I'm curious, is there any chance that they're actually adding this modification onto the proteins that you're implicating in the bottom right graph? Or do you think the function is fully independent of the diptamide biosynthesis pathway?
I guess if it was like a diptamide modification itself, I was expecting more that I would have seen similar changes in the protein expression profile across all of the mutants. So because all 7 of them are like required to actually synthesize the diptamide modification. So based on this, I'm thinking more that it's alternative functions.
Thanks.
Other questions?
So diptamide is required for elongation factor 2 function, or it just slows down translation?
So it's interesting. Diptamide's not actually like required for translation. Like these yeast cells I grow, they are fine without it. And so there's been like some more recent studies showing that like lack of diptamide can increase the amount of like frame shifting during translation. So I think that's one of the hypotheses.
Interesting. Yeah, the reason I asked is that your biggest hits are ribosomal biogenesis factors, and I know if you have issues in translation, that can like back up the production of ribosomes, and so then you're going to downregulate all your ribosome biogenesis factors. So I wonder if it's just like a buildup of ribosomes that aren't really working very well, or having a bunch of frame shifts and getting stuck, or something like that.
Yeah, I think it would be interesting to see if like certain proteins, like if their translation efficiencies are more effective because of lack of diptamide. So yeah, that would also be an interesting thing to look at.
Yeah, thank you.
Great. Let's thank our speaker one more time. Our last presenter, Yuji Wang, from Raggett Roberts Wood Johnson Medical School.
Hi, good afternoon, everyone. My name is Yu Jue. I'm a manager of the metabolomic core facility in Rutgers University. And today, I'm going to talk about the natural abundance correction from dual isotope tracer experiments. So first of all, I want to just introduce what is natural abundance correction and why we need to do that. So here is an example. We use 2-carbon-13 labeled acetyl CoA as a tracer and mix with the non-labeled species. And when we measure these species in mass spec, what we could see both the non-labeled M+0 and the labeled M+2 as the tracer. But meanwhile, you could still see some other unexpected species like M+1, 3, and 4. So the reason you see these species is because, remember, the stable isotopes are naturally present, right? So there is like 1% of chance of carbon-13 and 0.2% of the chance of oxygen-18 present. Therefore, because you have such a large number of carbon and oxygen molecule, so the chance to get a carbon-13 due to natural abundance is extremely high. And therefore, you could not simply just use this raw data to do the downstream analysis, and you need some way to correct this data first. And that's what we call isotope natural abundance correction. And the way you correct this data is really depends on the resolution. So no matter how fancy your machine is, you will always see two situations. The first situation is that your resolution is not enough. Your resolution is enough to specify carbon-13 and nitrogen-15. In this case, the nitrogen-15 do not actually interfere your observation, and you could just leave this data alone. The second situation is your resolution is not enough to resolve these two species. And in this case, the nitrogen-15 do interfere with your carbon-13 observation, and therefore you do need to remove this part from your observation. So as a core manager, we see more and more of our customers nowadays start to use dual isotope tracers in their experiment, and they ask us to do similar things for their data. And we realize there are no such useful tools so far to do this job. So that's why we decided to develop the Agar tool, which is a resolution-dependent algorithm to deal with the dual isotope tracer correction. And when we developed this software, we realized that unlike the single isotope tracer problem, where you could always get an answer from your data, in terms of this situation where you use two isotopes at the same time, you may not always get the answer, because this kind of problem actually depends on your resolution of your machine. So if your machine don't have a sufficient resolution that could resolve the two isotope tracers that you use, then you will face a situation like this. So you will, for example, here, one carbon and 0 nitrogen labeled species and 0 carbon 1 nitrogen labeled species could not be distinguished, and they will be observed together as M+1. And in this case, you will need to use only 5 observations to solve 8 unknowns, which is mathematically not possible. So that's why you need at least a sufficient resolution to resolve the carbon-13 and the nitrogen-15. Then you will get enough observation to solve the problem. So we have developed Agar tool, and we tested this performance using a NAD spectrum observed under 750K resolution. And here is the expected result. And our correction showed a very good correction after the performance of the software. And compared to our software, we also compared the other two available tools claim to do this data, but actually they failed to recognize a lot of species due to natural abundance curve. So we have made this software 100% free online. And if you are interested in doing dual isotope tracer in the future, you are welcome to learn more from our website. Yeah, thank you.
Wonderful, thank you. Do we have questions? Go for it.
I'll use your microphone.
Yeah.
It's quicker. So can you tell me, you said in your core facility, lots of your customers want to do dual tracers. Can you tell me what their applications are typically?
Yeah. So most of the application is, so when you have a complicated pathway that requires two entry of the substrates, then you could label one substrate with the nitrogen and another one with a carbon-13. Then you can do a flux analysis. And this way, you avoid to, of course, you can use a parallel labeling strategy, but it will be more complicated. If you use the two tracers at the same time, you will save some time and save your experimental effort, and you can get your result quickly. But of course, you give us more labor to do the correction. Yeah.
Awesome. Thank you so much. This was a great session. Thanks to all of you for the attention, and let's thank all our speakers one more time.